So we're in your biology and we're in chapter 12, section 2 on infectious diseases. So we are going to just travel through this chapter. We're going to go through a lot of different infectious diseases, a lot of information, and you can go back and study it, but I'm just going to whiz through. So infectious diseases are caused by pathogens. So here's your word pathogen. So what is a pathogen? Well, a pathogen can be bacteria and viruses are the most common pathogens, but it also could be a few other things. You know, pathogens, of course, need conditions of warmth, moisture, and nourishment, and they need a body host or a human host, or pathogens need to live off someone. That's a pathogen, off a host. There's other pathogens that include protozoa. They're tiny single-cell animals called protozoa, fungi, and some algae, and certain worms. And there's a few other creatures that are pathogens. So pathogens cause infectious diseases. So the diseases can be grouped in two different ways, not communicable and non-communicable. Communicable disease are passed person to person, either directly or indirectly. You know, either directly and they cough or something, or indirectly, you know, off their pencil that you're touching. You know, that's indirectly. So those are communicable. Those are things like colds and flus and AIDS even, and the plague. There's quite a few of them. And there's also non-communicable. Non-communicable cannot be transferred person to person, but they're still infectious diseases, like tetanus would be one. Tetanus is an infectious disease, but it is non-communicable, not contagious. You know, can't be transferred to another person. So let's talk right here. I'm going to zoom in to these diseases here. Let me go up here so you can see. Basically, bacteria diseases are like pneumonia, diphtheria, tetanus, tuberculosis, leprosy, are caused by the pathogen bacteria. Viruses are cold, flu, rabies, smallpox, yellow fever, polio, and viral pneumonia. So quite a few of those. Protozoa, we have protozoa, African sleeping sickness, and amoebic dysentery are two of those, but a very common protozoa that infects so many people of the world, on all of the tropical areas of Africa and South America, is malaria. Malaria is a protozoa. And then fungus. The fungus are like athlete's foot, ringworm, and they're called, this word, mycotic, right there, mycotic. Mycotic are fungus. And then worms. Worms, roundworms, hookworms that come in your feet, tapeworms that are in your intestines, flukes are in your blood. And these are helminthic, right here, helminthic diseases. So helminthic, just think it's helminthic or words, kind of, you know, worms, and then mycotic are a fungus. Those two things you need to remember. So let's go on, because we're going to talk about bacteria now. Probably for the rest of this video, we're going to focus on bacteria and viruses. So let's go here. So I'm going to go over to this picture, and this is the structure of a bacterium. Man, very unusual, isn't it? And you see, okay, bacteria are unicellular, means they're only one cell. They're widespread all over the place, you know, and they're different. There's different bacteria that are on plants and different bacteria on animals. Their structures. Now remember, there's two different kinds of structure here of these cells. Eukaryotes are algae, fungi, protozoa, all those, because they have a cell membrane, and they're called eukaryotes. You know, and they also have a nucleus, a bound nucleus, and they have organelles inside. So they're like normal cells. But a bacteria is not like a normal cell. It's called a prokaryote. A prokaryote is basically does not have genetic material in a nucleus. It has just genetic material just kind of clumped in a region. Looks like a region, but it isn't really surrounded by an organelle or organelles. And basically the DNA is very unique because they only have one chromosome. The DNA is circular. As for what else they have plasmids, and these are small DNA circles, plasmids, and they contain genes that make resistant to antibiotics. Bacteria can, become resistant to antibiotic, and that's the bad thing, because antibiotics usually can kill bacteria, but now some are becoming very resistant. So the cell walls are made of peptoglycan, I should say. It's not made of cellulose like a plant. And some have have a slime layer around them, like Streptococcus mutans have this little slime around them. Most of them are in these tough capsules that protect the bacteria from drying out. And some are mobile, and that means they can move around. And some have flagella. And flagella are what can make them move around usually. And you see, and they're driven by these flagella. So let's look at it right here. All these things I've been talking about. So this is the bacterium. So here we have. Let me go up here down a little bit there. Oh, there we go. Here at the top, we have there's the ribosomes in there. Ribosomes are their protein factories, and then you have the outer cell layer here, outer cell membrane, and then you have the peptoglycan layer underneath. So 2 layers there, the outer cell membrane, and this is both these together are the cell wall of it. And then there's the inner cell membrane. So you have look, it looks like 3 parts. You have the outer cell membrane, peptoglycan, and then you have the inner cell membrane. This here is the cytoplasm, and it contains enzymes and other cellular components. And then you have the nucleoid. It's kind of not a nucleus, but it's just, see all these right here together, that little area, or this area here, they're kind of scattered around there, contains the DNA molecule. And there's the basal body there, the flagellum, that's how they can move with these hair-like things that moves them around. And these are the pores in the outer membrane. That is a weird creature, don't you think? That bacteria. There's bacteria come in different shapes. This one looks like the bacillus shape, the bacilli shape, I should say, kind of that rod shape, but they come in quite a few different shapes. So when I think of different shapes, let's go up here and we'll see these shapes. Bacterial shapes. So the first shape up there is called the cocci. The cocci are like spherical, and they're clusters and chains. Here's how they're tiny. They come together, cocci. And then you have the bacilli, and that's what we talked about right there. They're kind of rod shaped. And then you have the spirilla, and the spirilla are spirals. So what about these? Well, they're very, very pathogenic, so they infect, that's for sure. The pathogenic cocci, they clump up like grapes, and there's quite a few, like Staphylococcus aureus is the most common. And staph can be food poisoning. It can also have staph infections on your skin, so not a good thing. Staphylo means bunch of grapes. So you know that Staphylo is going to be a cocci. It's going to look like little grapes. There's also Streptococcus pneumonia, which is a really a hard pneumonia type that infects bacterial pneumonia, I should say. And strep throat, scarlet fever, those things are also under cocci. Strepto means twisted in Greek. So it's kind of a weird twisted in Greek is kind of a different word, but strepto. Other kinds of staph can have boils, carbuncles, even meningitis and gonorrhea. So quite a variety of infections. And then we have the bacilli there. The pathogenic bacilli are mycobacterium. things like tuberculosis, mycobacterium, leprosy, salmonella, food poisoning, typhoid fever. And then we have one that's called E. coli. E. coli, lots when you think of E. coli, you think of like manure, you think of feces, because that's where a lot of E. coli are. So when you have food poisoning with E. coli, you think, oh, the sanitation was terrible. Somebody's waste products cut into the food, E. coli. And also bacilli botulism, which is the poison, actually the poison from botulism is the most poison thing you can ever have. And diphtheria, plague, tetanus, they all come under the pathogen bacilli. Now the spiral one, the spirilla, they're only single cells. They look like little corkscrews, as you can see there, and they produce, but this corkscrew is kind of producing a bacteria of certain things like Lyme disease and what else? Tetanus and botulism. When I say that, I think you think of the spiral that can be aerobic or anaerobic. Well, that's aerobic means it needs oxygen. Anaerobic means it does not want oxygen at all. And there's some kinds of anaerobic, like botulism and tetanus. Tetanus is when you get a cut, a puncture wound, and it's anaerobic because it goes straight into your body there, and it doesn't get oxygen, and this tetanus grows and causes lockjaw. It can kill you. So it's so important that you have a tetanus vaccine, tetanus shot, I should say. That's what I always think. Get your tetanus shot. Well, also botulism is in canning, and it's dangerous food poisoning, and it lives in those cans that have no oxygen. So there's some that need oxygen and some that can't have oxygen. So it's a variety there. So heterotrophic and autotrophic bacteria. So let's go and next. See, maybe we'll see some. Okay, I'm gonna leave it there. But we have heterotrophic and autotrophic bacteria. Autotrophic means that they're a self-feeder. The bacteria can actually support itself and make its own food. Now, most of these we've been talking about are heterotrophic. They're unable to make their own food. They're called other feeders, like parasites and saprocytes. So these parasites that live, and they live in some areas. And some of these, and they're not all bad. Some of these bacteria are beneficial. Did you know in your intestines live this beneficial bacteria that helps your intestines? Your intestines need them. In fact, it fights other bacteria. So, but that's the important thing about some are good and some are bad. Autotrophic bacteria contain chlorophyll, and they have photosynthesis. They go through photosystem, but they lack a chloroplast. So their photosystem's a little bit different. They use hydrogen sulfide and toxic chemicals in this chemical synthesis, not synthesis from the photosynthesis from the sun. And the chemical synthesis needs iron and sulfur and nitrogen, and it takes the place of sunlight. So it's very, very unique for bacteria. Reproduction of bacteria. How do they reproduce? Well, they reproduce by binary fission. That means they're asexual. They don't have anything to do with sexual part. Binary fission means they just basically cut off and make another one, right? One cell splits into two cells, and they grow in a narrow range of heat, light, and moisture, although some can grow at all different temperatures. At the optimum condition, bacteria can reproduce every 20 minutes, and 2 days you can have them reproducing 27,000 times in 2 days. So, and ricochet is they reproduce only inside living cells, and that makes them parasites. And there's certain kinds of bacteria that are in insects and spiders and crustaceans too. And then there's typhus and Rocky Mountain spotted fever, and they're transmitted by ticks and mites and insects. So insects can be vectors for bacteria, and transport all kinds of these diseases, bacterial diseases. So when you think of Rocky Mountain spotted fever and ticks and mites and insects, especially ticks, can give Lyme's disease. So conditions. So let's talk about the conditions a bit here. So, and then I'm going to go into that right there. Reaction to unfavorable conditions, it says here. So what do you think when they react to unfavorable conditions? Well, they produce these endospores. So when unfavorable conditions occur, such as extremes of heat, cold, or dryness, and many types of bacteria protect themselves by becoming dormant. And they do this by this internal life support capsule called an endospore. And it's really weird, but endospores, they can go dormant for years even. So, well, you know, when you think of endospores, even before I think of endospores, bacteria can live, some bacteria can live in really freezing places, and some in the deep sea hot springs at 500 degrees Fahrenheit. They found bacteria in hot springs that hot, 500 degrees. Also, under a lot of pressure, they found bacteria under 27,000 Pascal, kPa or PSI, 4,000 PSI. That's a lot of pressure. And some thrive in brine, which is salt water, and some need strong acids, and some need to be bases instead, neutral or bases, and some require oxygen, some not oxygen. They found bacteria on top of Mount Everest and in the bottom of the Challenger Deep. So many areas. What else? Bacteria. Unfavorable, sometimes when they have unfavorable, they develop this endospore. The weird thing about endospores, this thick wall capsule around them, right here, which looks like the endospore there, it germinates, and so it's very difficult to kill an endospore because there it's protected. And some of these endospores that can survive, they said here, 1,000 years, thousands of years. They found anthrax with its endospore in ancient tombs of Egypt. And you know what? They've even found some of these endospores in times when they thought were before the flood, Diluvian, pre-Diluvian, before Noah. That's a long time ago. So let's look here. We have an endospore. It says bacteria under favorable conditions here, and it goes under unfavorable conditions, bacteria packages of vital machinery to inside the endospore. There he's putting that stuff in the endospore. The endospore survives harsh conditions that would destroy protected bacteria. And on here you see bacteria breaks out of the endospore and forms a new cell wall when conditions become favorable. So that's the endospore, bacteria. So let's go on to the next here. Bacteria and disease. Let's see. I was going to go to quote, but I thought we haven't. Yeah, here he is, Louis Pasteur. Let's talk about Louis Pasteur in the midst of here, because it's pretty important, because he was one of the first studies of bacteria. Louis Pasteur lived 1822 to 1895. So, you know, we're talking about, you know, over 100 years ago, right? So it would be actually almost 200 years ago. He was French, and what he did, he basically saw that bacteria was used. He thought that couldn't see it, but that something was causing wine to turn to vinegar and ferment, and he thought it was bacteria. This bacteria, is bacteria the cause? Is this cause of bacteria for that also the cause that causes our body to be sick? So he thought about that. He said, are these bacteria, do they cause disease? And so Louis Pasteur, he made the germ concept of a disease, that every disease must be caused by a specific germ. Now, he didn't necessarily label them as bacteria, but he said germs. He called them germs. So he also put together pasteurization. Pasteurization before milk you had to boil milk before you drank it, because it could kill you because the bacteria are hazards in it. So what he did, we at a lower temperature, he'd heat up at 72 degrees centigrade, 161 degrees Fahrenheit for a few seconds to make milk safe to drink. And it would also keep the milk from spoiling really rapidly. And so that was part, and he realized that as he was pasteurizing these things, that there was something in there, a pathogen in there, and that would be bacteria. In 1882, this is a great story. So in 1882, let's move this up here. There he is in his laboratory. There's a fatal viral disease that attacked the brain called rabies. Rabies would cause mental disorders, delirium, death, and it was produced from a virus, actually, not a bacteria, but a virus. And so he thought if he could produce a weakened form to the virus, that he could possibly give a weakening form to someone, and they would develop, it wouldn't infect them, and he would develop basically these antibodies or resistance to the disease. So he thought this weakened, attenuated, and you can inject into a healthy animal, and doing this, the animal would produce antibodies against the virus. So this would be a type of vaccine that would make a person immune to rabies. So he didn't really test it on a human being until a 9-year-old, Joseph Meister, was a young man, and he was bit by a rabid dog, and he had rabies. Rabies would just kill you. So the mother pleaded to try to cure him, and so the treatment that he gave him through the rabies and this attenuated injection, virus hadn't even been discovered at this time, so he really didn't know what he was totally doing. But he gave Joseph this, and Joseph was cured. It was like amazing. So what an amazing story. So, and Louis Pasteur, he loved the Lord. He said, the more I know, the more I have faith in God. The more I learn, the more I trust God. It's amazing. So Louis Pasteur. So let's get you a little bit, talk about Robert Koch's postulate. Let's go back. I skipped that over that. Let's go back there. Applying Koch's postulates. Here we go. See if we're gonna put that up just a little bit. There we go. So Robert Koch was a German physician. He lived in 1843 to 1910. So similar to the time that Louis Pasteur lived a little bit earlier. And he did experiments on anthrax. And what anthrax was, it was a killer of sheep and cattle, a bacteria that would kill them. And so he was going to develop techniques so he could study this bacteria. So he was the first great bacteriologist or bacteriologist. So he was a scientist that studies bacteria. So what he did, he put some postulates on how you would study. So basically these are it. So the microorganism must be observed in the blood or tissues of the infected animal or plant and must have a reasonable relationship to the disease symptoms and lesions. So he's basically going to use this rat here to as a disease to see how the disease reacts in a rat. And then the next is the organism must be isolated from the diseased host and grown outside the body in a pure culture. So he put these cultures of bacteria and grown them in a culture. And then number 3, a portion of this culture must be injected into a second, previously unaffected animal or plant to see if it causes it again. And number 4, symptoms and lesions similar to those in the first host must appear. So that's how he said, if really these bacteria are causing disease, you must go through these 4 things. So pathogenic bacteria would be there. So And he also proved Pasteur's germ concept. So Robert Koch was his name. Robert Koch applying Koch's postulates. So pathogenic bacteria, what do they do? Well, how do they react? They steal cell nutrients is the simple way. They go into a host and they steal the nutrients of the cells. They also produce toxins and damage cells, like tetanus. Tetanus produces a toxin, and gangrene is another one that produces a toxin that breaks open body cells. So these toxins, and then after the toxins break open the body cells, the bacteria feed upon those cells that are broken up. They also just basically turn the tissues into a battling ground. You know, the whole a battleground for the immune system and invading bacteria. The body cells are caught in the crossfire, which means the person would have a lot of swelling and scar tissue. And some cause the immune system to malfunction and to have more problems going on. So bacteria can first steal cell nutrients, produce toxin, and just turn all the tissues into a battleground. So bacteria. So I'm going to stop there, and then we're going to go on to viruses.